Tom is the CSO at Tumor Bioscience, which is a clinical stage gene and cell therapy company focused on curative intent in solid tumors. And he's also the founder and CEO of Empyrean Therapeutics, based here in Cambridge, developing genetically engineered cell therapies and underlying gene editing technology platforms. A large part of his research is focused on establishing CRISPR-modified T cells for translation to the clinic and investigating how antigen-specific T cell responses can be improved by targeting novel immune checkpoints. He holds a PhD in molecular immunology from the University of Cambridge, and today he's going to be speaking to us about a novel cell therapy for COVID-19 and potential future pandemics, virus-induced lymphocytes. So Tom, thank you very much indeed for speaking to us today, and I'll hand over to you.
Great. Thank you, Jeremy. And obviously, thank you for the invitation. Can you see the screen okay?
Yep, all good.
Excellent. Okay, perfect. So, yeah, so what I want to talk about this morning, good morning, everyone, it is a brief talk. Hopefully it shouldn't be too long. And it's from a piece of work that we did during the sort of peak of the pandemic. Like others, we were looking at, how we could pivot our technologies in sort of cell and gene therapy into anything that could be useful, obviously, for treating the underlying disease, obviously, for COVID-19. And in Tumor Bioscience, what we're focused on is solid tumor curative intent T cell therapy. So we envisioned an opportunity whereby we could actually see about developing a cell therapy platform for virus-specific T cells with the objectives of taking SARS-CoV-2 patients that would be suffering, obviously, in the ICU with sort of severe form of the disease, isolating T cells from the blood, and then, could we actually, over a very rapid time point, in 7 days, actually expand those T cells, both CD4s and CD8s, both in quantity and quality, with the aim to then reinfuse back into the patient, with the idea that if we could potentiate the immune response through a sort of therapeutic or clinically relevant number of T cells, can we sort of augment the response that these patients are obviously their own immune response during this time? And this was based upon some indication we had. Some of our staff, our CEO, for example, went back into the COVID ICU at New York Presbyterian during the peak of the response and was treating patients and saw kind of their time course in the ICU, especially those sort of intubated patients undergoing sort of, pulmonary distress, about how long they were in the ICU, and particularly, obviously, their T cell responses that were widely reported at the time in terms of, low T cell numbers, lymphopenic sort of early stage disease, and then an increase in that response. And it would seem that there was early evidence at the time mounting that the actual T cell repertoire and the T cell response and the potency of that seemed to correlate potentially with some sort of good prognosis of the disease. So what we did is we looked at our platform that we have now in the clinic in terms of our tumor infiltrating lymphocyte expansion immuno-oncology platform, which is the idea there is that we take tumor fragments from cancer patients. These are late stage 4 patients. We isolate the tumor fragments. We isolate the tumor infiltrating lymphocytes. And then through a standard sort of rapid expansion protocol, we look to expand them to a very large number, sort of 1E10 numbers of neoantigen specific T cells, which are obviously specific for the patient's own tumor. This takes several weeks to do because we need obviously very high numbers, but all of the underlying methods are focused on the amount of cytokine to use, the expansion vessels, the T cell density, as well as obviously the antigen that we're stimulating with, which in this case is overlapping pools of antigenic peptides from exome sequence tumor samples. So we envisioned, can we take some of this and in parallel try and develop this and shorten the time period just over to days rather than weeks, so in 7 days, but take a similar sort of approach where we have an antigen-specific expansion of these cells, develop a mode of expansion beyond just an overlapping peptide pool, and then actually try and expand and enrich that antigen-specific population, as opposed to just sort of generic expansion of those T cells. So it was really focused on the T cells that were responding to the SARS-CoV-2 antigens. And this is where we sort of teamed up with ProImmune that were developing reagents at that time for the detection using sort of pentamers and tetramers of SARS-CoV-2 specific T cells. So this obviously offered us a route that if we could detect them, perhaps then we could actually antigen specifically stimulate them as well. So We're also interested in sort of the comparative here between what patients were receiving at the time in terms of standard of care in the ICU, where they were getting convalescent antibody sera from convalescent patients in a sort of passive attempt to engender some humoral immunity in a transient way, whereby this was showing some level of efficacy, of course, that antibodies, that could be delivered to these patients from this convalescent sera seemed to have some potential promise in efficacy, although kind of it wasn't as robust, I think, as people were hoping. And of course, the underlying cellular component of a T cell mode of action in terms of a cellular therapy was obviously very attractive because of not just the ability to engage and engender the humoral response by B cell activation, but then potentially a longer lasting immunity as well. So just to sort of show you what we borrowed here from our oncology platform, this shows our schema for sort of TIL expansion in the clinic at the moment, which is in a phase 1/2 trial in the US. And as I said, this usually is over several weeks. And a lot of this is because we have to isolate the tumor fragments exome sequence to identify the neoantigen profile of these patients, then produce very quickly pools of peptides, and then a co-culture system using antigen-presenting cells derived from patients' dendritic cells. We then actually sort of do the antigen-specific amplification in a presentation assay. But once the cells are activated and we get outgrowth of the neoantigen-specific T cells, then it's all around about the methodology of how quickly and robustly you can expand the cells in such a way that they're healthy, they expand and they're activated to high numbers, but obviously we don't want them to go into any exhaustive state where they're not going to have the functionality that we intend when they're obviously re-administered and re-delivered in vivo. So this is something that we've worked with the NCI over numerous years to develop quite robustly. And this is technology which in the past has had curative responses in solid tumor. So what we wanted to do is take the aspects of that, particularly in terms of the high cytokine doses, which seem to be important at the early stage of this expansion, such as higher levels of IL-2, as well as certain flasks. In this case, this G-Rex gas permeable flask system was quite important because it just allowed us to sort of kickstart the cultures in such a way whereby at very sort of high densities in small volumes of media, the T cells seem to enter into that proliferative sort of exponential growth stage in the first sort of 24 to 36 hours, which was obviously important then for that outcome at day 7, that those cells, their expansion was potentiated. And this shows you the scheme here at the bottom of our clinical trial. And of course, we really want this first 7 days. And again, 7 days, obviously, this remains to be seen if this is actually clinically relevant. This is something we hope to obviously test, and I'll explain more about that. But of course, again, this was really based upon what we were seeing in the ICU. And of course, a 2-month-long expansion therapy was not really going to help anyone suffering from this, quite severe forms of this disease in the ICU. So the platform we built was based around micro artificial antigen presenting cells, which effectively are simply just paramagnetic beads, which we then conjugated onto them the MHC class 1 pentamers with peptide specific for SARS-CoV-2 immunodominant antigens, which have been identified around the time for expansion and activation of CD8 cells, or MHC class 2 restricted tetramer antigens here. So tetramers for the engagement of and amplification of CD4 cells. And using a bead-based system, and we're quite familiar, obviously, with the sort of Dynabead CD3, CD28 expansion of T cells. We wanted to kind of emulate that, but obviously in an antigen-specific rather than a pan-T cell activation way. So we also went for an approach which, whereas others, favor the idea of soluble peptide, which seems to work quite well, and this is something that we're now comparing. But we wanted to add this CD28 co-stimulation because we felt that that was important to obviously get the phenotype of the cells that we want. So the highly active memory-like cytokine-producing T cells, but also, could this be a key to sort of that early stage amplification? So anyway, this is what we went ahead with. And as you can see, this is the schema for the experiments here. The isolation of this from PBMCs, the T cells, and then that 7-day expansion in the cytokine culture, and then the flow cytometry readout that we did. And we stuck with at the time the most sort of potent that we could see, MHC-1 restricted SARS-CoV-2 antigen that was then obviously in the form of a pentamer. So here's showing in the same time we looked at in an orthogonal system just to show that this could be achieved is that, and the other thing was obviously getting convalescent PBMCs from SARS-CoV-2 recovered patients was quite challenging. I think everyone in the world wanted those samples, especially for the isolation of sera for treatment. So we also developed and optimized this platform using CMV infected individuals. These are previously CMV positive donors, isolate the PBMCs. And you can see here, again, using pentamer and tetramer reagents that are then fluorescently conjugated, we could detect antigen-specific cells in these PBMC samples, either using this PP65 class 1 antigen or the GP215 class 2. And you can see low-level presentation of these T cells. And then after 7 days, in the presence of what we call these Viper particles, these artificial antigen-presenting cells, we could see a significant augmentation and expansion of those populations, which was quite nice in these CMV samples, both CD8 and CD4. And this was, a robust sort of enrichment. And then we focused on coupling that enrichment with an expansion, because this all really comes down to a numbers game. Can we get enough cells through enrichment and through sort of total pool expansion? We did some further optimization. So here you can see, again, this is the actual particle itself. We played around with the density of the pentamers and tetramers and the ratios of anti-CD28. And you can see here, what we've done is that we've looked at the dosing, which was surprising to us, that they seem quite potent, that you can go down to a ratio of 1 particle per 20 T cells and actually get the best augmentation and potentiation of the expansion. So we sort of went on the lower end here. And we also looked at the density of CD28. And interestingly, we found, and this has been demonstrated before when we started looking to the literature, is that when you have a system where you are obviously stimulating in an antigen-specific way through the T cell receptor and then bringing in that CD28 co-stimulation, the CD28 really does help to sort of potentiate that expansion. So we actually found that a higher density, a ratio of where we had sort of 30 molecules of CD28 for every pentamer or tetramer, seemed to work a lot better as well. And we've actually pushed this further now to sort of tweak and optimize a little bit more. So then we switched to PBMCs from our convalescent patients. And you can see here, this was our first sort of that experiment where we took, this is, at the time, we were focused only on class 1 amplification of CD8 cells, because there really wasn't any good proven out CD4 expansion reagents in terms of MHC2 restricted antigens. So we initially focused on the idea that we could expand CD8 cells to a meaningful number. And you can see here that at day 0, when we took PBMCs from convalescent patients, we could barely detect any cells. And this was sort of the hallmark of what we saw, no matter who the patient was or the presentation of their disease, or even the time after recovery. But encouragingly, what we saw is that after 7 days, in the presence of the particle, we saw the emergence of this population to a fairly easily detectable number. Obviously, without particles, just in cytokine and expansion growth media, we could see an expansion of the population in total, but no sort of antigen-specific cells emerging. And you can see here, this is shows you some of the profile of the patients that we were isolating cells from. These were all PCR positive in terms of the presentation of their COVID-19, as well as IgG and IgM positive serological results. They were symptomatic, and we specifically tried to find those that were either hospitalized or had severe symptoms, again, to try and sort of closely match, how this would be in the real clinical setting, should this be a mode of treatment. And the most important thing is that obviously, and potentially a limitation that would require overcoming for this, is that prior knowledge of the antigen or the dominant antigen, which was quite impressive how quick this was developed for SARS-CoV-2 and the COVID-19 pandemic, does require obviously MHC matching of these patients to their particular Viper particle or the antigen that we'll be using to expand them. And we've now actually expanded this into other HLA types and antigens. So we actually envision this could be a panel of reagents that could be based upon the MHC type of those samples and patients, that we could then pivot and use different different particles depending on their MHC. And we actually now have a process in place to quickly MHC overnight type PCR type the samples in process as well, so we can actually tailor and match up the correct reagents to the correct MHC. And here you see some of the expansion results that we saw on average for that first sort of primary development of this. We saw about 1,000-fold expansion. And this was both whether we actually tried this with CMV specific or SARS-CoV-2 specific reagents and corresponding patients. So you can see here, this is the actual percentage of CD8 cells that we could increase, the numbers in terms of the input of a few 100,000 cells and what we ended up with. But perhaps most importantly, this was the fold change. And should this scale, then this allowed us obviously to calculate what this means in a sort of meaningful setting with actually starting numbers that are sort of clinically relevant. This is SARS-CoV-2, and this is the CMV PB65 CD8 expansion here. And again, even though we had much higher starting numbers because these cells were much more prevalent in these recovered individuals, we saw about the same kind of fold expansion, and we've now optimized this further, so we're actually getting closer to about 5,000 to 10,000 fold increase as well. And again, most of this comes down to simple sort of T cell handling and keeping the cells happy and expanding. So then we went on to analyze the phenotype of these cells, because obviously quantity is one thing, but quality is the other. And we wanted to see exactly after 7 days, which is, reasonably early in these ex vivo cultures, certainly earlier than we analyze and look to sort of affect in our oncology cell therapy, is that what do these cells look like in terms of their activation profile and their functional responses. And we saw largely at this earlier time points that they looked quite similar in their profile and expression of activation markers to the non-antigen specific population that was obviously proliferating and growing in those cultures due to the cytokine response here. So expression of 4-1BB, for example, and the pentamer positive and negative population for OX40 and other co-activation markers seem very similar. Quite a spread between patients, seemed to be a lot of donor variation. And we also looked at co-inhibitory receptors to see if there was any hallmarks of an exhaustive phenotype in these T cells, particularly in the case of PD-1 and lag and TIGIT expression. But again, they look quite similar to those antigen negative populations as well. So there's nothing that suggested that they were particularly exhausted or T cells that we would potentially be worried about. But of course, the main thing there then was, what is their phenotype here? So in terms of the memory formation, we had looked because we were interested in seeing that maybe one value here of obviously driving an activation response over 7 days of these T cells is not only are they going to be able to respond to the viral pathogen when they're introduced into the patient, but also, do they have the right phenotype that they could perhaps persist, not just activate the humoral arm of the immune response, but, are they going to be long-lived and allow sort of recovery over the days and weeks that's necessary? So we looked at using standard sort of CD45 RA-RO and CD62L staining in, again, the pentamer positive population here. So these are the antigen expanded cells at day 7. These are the non-specific cells at day 7. Just to see what difference there are in terms of their naive versus memory formation. And we did see, perhaps not surprisingly, that the cells that are antigen specific and robustly expanding have been driven to that more memory-like CD45 RA low population. And when we drilled down into looking at the T effector memory, which we thought was a good indication of function in that regard, again, we saw that this was an increase in that proportion. This is showing a nice result that from the CMV PB65, and we saw the same thing as in the publication that we put out with the SARS-CoV-2 as well, is that it seems to be a general shift at day 7 to a more sort of memory-like, pseudo-memory-like phenotype. And then we looked at the polyfunctional pro-inflammatory cytokine expression. Again, in the absence of doing any functional assays, looking at the ability of these cells to clear virally infected cells, for example, in a sort of cytolytic assay, or any other in vivo models at this stage, because this was, quite a quick study to sort of see what the minimum sort of phenotype we could see if there's value here, is that we wanted to see is that are these cells producing what we would term sort of pro-inflammatory cytokine. And we certainly can discuss more about whether there's value, obviously, in T cells in the pro-inflammatory state in terms of augmenting this disease as opposed to hindering. But it would seem, obviously, that these cells that would go into these patients that would then, help to potentiate the immune response, as well as sort of recruit other B and T cells. We think that this would probably have the correct phenotype, and obviously this remains to be seen and something yet to be tested. But what we saw again is that when we looked at the antigen-specific pentamer positive population, and we looked at interferon, gamma, TNF-alpha, and IL-2 responses, as you can see here, is that even though the negative population were definitely expanding, as you can see from those earlier numbers, is that we were getting 1,000-fold enrichments of the cells. Not surprisingly, in higher cytokine, is that they weren't activated to the extent where they are actually producing cytokine in a way after challenge, which again just shows that, as expected, is that these aren't antigen specific. So a re-challenge with the SARS-CoV-2 antigen, either in the format of a Viper particle to again stimulate the T cell receptor, or just by soluble antigen activation, we could actually find that this led to upregulation and expression of these cytokines. And this, when you look at this, obviously in a polycytokine response, you can see here in the antigen-specific population that it was also the increase in the number of and proportion of cells that were expressing more than one cytokine, also expressing 3 cytokines as well here. And shown in the numbers is that this was probably as expected, is that you would have the kind of response that you would see with a generic sort of dynabead stimulation, whereby you're doing that quite strong and quick activation through the T cell receptor with the co-stimulatory signal as well. So when we put this together effectively, and what we're kind of working on now is, how does this scale? If the phenotype of these cells are obviously clinically relevant, and when you look at the response here in terms of how this can be scaled to the number of cells that could be isolated routinely and safely and ethically from these patients, starting from the number that we would expect within the blood. And it remains to be seen, what is the number of starting virus-specific T cells in someone actively undergoing a viral response, be that to COVID-19 or any other viral pandemic that we might face, is that obviously, again, this numbers game and this rapid expansion means that we can hopefully just expand this into some sort of rational way, which is scalable and predictive, to get to the numbers that we envision here. So again, this was for the purpose of illustration, that 3.5 times 10 to the 9 virus-specific T cells, isn't far off to when we compared and contrast this to other therapies in which T cells are delivered in an antigen-specific way, either in the setting of solid or liquid tumor, or other responses here. So this may well be, something that could be certainly of additive value to these patients, especially those that might well be in the lymphopenic stage of this, whereby, for whatever reason, their T cell numbers remain quite low, and this could be a real way to augment that, so that they have a real good shot, obviously, engendering that T cell response. And finally, we're looking now in terms of future work, obviously optimizing that a little bit more, but it will be exciting to see if this could actually be delivered in allogeneic settings. So obviously this autologous requires patients to come in, cells isolated. It's always tricky with the timing, as you can imagine, but MHC matching is certainly doable and part of the process. So perhaps there is an idea for future sort of cell therapies for pandemics where you could actually take the known MHC haplotype of these patients, which could be identified quite quickly, and then if you have expanded T cells already, of that right MHC, as we and others have done, then perhaps this could be something where it could become much more of an effective short-term therapy that doesn't require obviously this cell therapy manufacturer, which would only work if there's obviously enough time to do that. So I'll end there, and just to acknowledge obviously the people that did this. This was a small project that we did, but involved a collaboration between scientists at Tumor as well as our collaborators broadly on our immunovirology programs at Harvard, and our clinical team at New York Presbyterian as well, and obviously ProImmune for helping us identify the actual antigens and reagents for this. So thank you very much.
Fantastic. Thank you very much indeed, Tom. I have a quick question, if I may, just to kick things off. It was just in relation to maybe the combination or use of combination of different antigen-specific, viral-specific T cells. Is that something that in the allo setting you're thinking of combining a range of immunodominant epitopes rather than just looking at a single epitope?
Yeah, I think that's a good. Yeah, I think so, because if it's a well-established disorder, I know, for example, for CMV, something that's well studied and there's immunodominant epitopes out there, and the immune response has been quite well worked out, then potentially not. But for emerging pathogens, and we certainly saw this, with SARS-CoV-2, is that papers were coming out that would suggest the immunodominance and the sort of potency of certain antigens, and then other studies that would come out and sort of show that there's much more effective ones as well. So perhaps at that stage, and definitely sort of patient variation, is that you could go with a broader approach where you do mix and match, and it also could mean that you don't have to know a priori the actual T cell repertoire of that patient. If there's panels that you could actually start to expand with, then yes, you have the best shot, I think, in the shortest time of sort of raising and potentiating the kind of T cells that are already there in that patient without knowing exactly the dominant immunogen that you'd need.
Yeah, thank you. Any other questions at all? If you want to raise a hand or unmute and ask. Okay, I think you mostly answered everybody's curiosity there, Tom. Thank you very much indeed. I really appreciate you joining us today for your presentation. That's great.
Thank you.
Okay.